Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So we were discussing about neurobiology, in which we discussed about the reticular theory given by Camilo Golgi and the neuron doctrine given by Santiago Ramanujan. We discussed about the principles that were given by Santiago. So we discussed about the principle of dynamic polarization, the principle of connectional specificity. Then we also discussed how he observed the structure, how he observed the nervous system using the Golgi stain, and by the way, also used methylene blue as a stain, and then how he gave out the two theories, like two principles, and then he also gave out the different types of neurons based on the structure. So he differentiated, he characterized the neurons, he classified the neurons based on the structural properties into unipolar, bipolar, pseudo-unipolar, and multipolar neurons. So he categorized them based on how many processes a neuron has. So we discussed about histology of nervous system. So we discussed about structure of neurons, and we discussed about the types of neurons, types which are based on the categorization which is based on the different properties like the structural property, functional classification. Then we also discussed about the neuroglial cells. Today's topic of discussion is myelination. So when we were discussing about structure of neurons, we learned that the axons of the neurons, they are wrapped by the myelin sheath. So myelination means wrapping of myelin sheath around axons of the neurons is known as myelination. Okay. So myelination is the wrapping of myelin sheath around the axons of neuron. So myelin sheath, this is a multi-layered structure. So this is a multilamellar structure of lipid and protein. So myelin sheath is multi-layered lipid and protein covering. Okay. So myelin sheath, we discussed that this is produced by the glial cells on the axons. Okay. So the neurons. So this is how axons, they are wrapped by the myelin sheath. So this is the myelin sheath. Okay, so the myelin sheath wraps the axon in the segments. So the part where there is no myelin sheath, that is called nodes of Ranvier. Okay, so this is how myelin sheath is present on the axon, and this myelin sheath, this is produced by the two glial cells, the Schwann cell and the oligodendrocytes. Okay, Schwann cell forms the myelin sheath around the axons that are found in peripheral nervous system, while the oligodendrocytes form the myelin sheath around the neurons, around the axons of the neurons, which are found in the central nervous system. Okay, this is the difference. So we discussed about this when we were discussing about the glial cells. Today we will be discussing about the details of myelination. So there is differences there about in the myelination in the PNS and in CNS, because two different glial cells are going to form that myelin sheath, right? So what happens is that the Schwann cell, it starts forming the myelin sheath during the fetal development. Okay, so the myelin sheath formation in the peripheral nervous system starts during the fetal development. So there's a mechanism. This is a step-by-step process by which Schwann cells form the myelin sheath. So Schwann cell wraps itself around the axon, and that wrapping is that is of 1 millimeter thick. Okay, so myelin sheath which is formed around the axons of the PNS, they are about 1 millimeter thick. So let me show you a diagram where it will be much more clear. So here you can see this is a Schwann cell and this is the axon of the peripheral nervous system. Okay. So what happens is that this Schwann cell, it has a nucleus and it has its cytoplasm. So this Schwann cell wraps itself around the axon of the PNS. So here you can see that it is wrapping itself around the axon. Okay. So this portion stays here and this wraps itself around the axon. So here you can see, and this is how it forms multiple layers. So here you can see this is step 1, this is step 2. Here you can see this portion is wrapping itself around the axon. So here you can see it is forming multiple layers. So once it forms multiple layers around that axon, that is called myelin sheath. Okay. So myelin sheath is nothing but the plasma membrane of the Schwann cell, which is wrapping itself around the axon. So as the myelin sheath is a plasma membrane of a cell, so I hope you will be that will the composition of myelin sheath will be easier to guess, right? So myelin sheath, it is the plasma membrane. So basically it is a lipid bilayer which is wrapping itself around the axon. So that is why myelin sheath is consisted of lipid. The 70 to 80% part is made up of lipids, while the 20 to 30% part, the rest of the part, it is made up by it is made up of some proteins. Okay, so myelin sheath is basically a lipid and protein covering the axons. Okay, so the other part, the cytoplasmic and the nucleus part, that forms the outer covering of the myelin sheath. So this outer covering is made up of cytoplasm and nucleus of the Schwann cell, and this outer layer is known as neurolemma. Okay, the outer layer of Schwann cell cytoplasm and nucleus is called as neurolemma. So this neurolemma is present in PNS, but it is absent in the oligodendrocyte. We will discuss why. Because neurolemma is the cytoplasmic as well as the like it's the cell body part of the Schwann cell. So that Schwann cell, which is residing its cytoplasm in only in the outer part as well as the nucleus in the outer part, that is forming neurolemma. And the neurolemma is the one which has the regenerative properties. Okay, and that is why the PLS myelin sheath has more regenerative properties than the central nervous system one. Okay, so let's discuss about it here. So as the so here let me write it here. The cytoplasm and nucleus is basically the cell body. The whole cell body of Schwann cell forms the outer layer, which is known as neurolemma. Okay, lemma means the layer, so neurolemma. Just hmm. So neurolemma is the basically it's the plasma membrane of the Schwann cell. That is why neurolemma is also known as sheath of Schwann cell. Okay. So as it is the sheath of Schwann cell, the Schwann cell is making the outer covering of the neurons. It is only present in the PNS. In the central nervous system, a single oligodendrocyte, it puts forth about 15 broad flat processes that spirals around the axons. So a single oligodendrocyte, it forms about 15, it put forth about 15 flat processes which spiral around axons. And this is how the myelin sheath is formed in the central nervous system. So the major difference is that a single Schwann cell is going to wrap a specific part of the axon, and then the other Schwann cell will wrap a specific part of axon, leaving the gap in between both of them, which is known as node of Ranvier. And this is how a single Schwann cell will so multiple Schwann cell will form the multiple segments of myelin sheath on a single axon. But on the other side, unlike Schwann cell, a single oligodendrocyte it just puts out a process which spiral around the axon, so which wraps the axon. So a single oligodendrocyte can cover multiple neurons, multiple axons of the multiple axons, okay, in the central nervous system. So this is the major difference between them. And due to this reason, when the single process multiple times, a single process of the oligodendrocyte covers the almost the whole axon, and that is why the nodes of Ranvier are very few, okay, compared to what we see in peripheral nervous system. So in the central nervous system, nodes of Ranvier are quite few because a single process, in many cases, a single process of oligodendrocyte almost covers the whole axon. Unlike Schwann cell, so this is the axon of peripheral nervous system, and this is the myelin sheath, okay, which is wrapping itself around the axon. Okay, so a single Schwann cell is making this a portion. A single Schwann cell is wrapping this part of axon. Other Schwann cell is wrapping this part, and the third Schwann cell is wrapping this part. And that is why it leaves multiple nodes of Ranvier in between two myelin sheaths. But unlike that, in oligodendrocyte, a single process in the central nervous system, a single process of oligodendrocytes spirals around the around the axon of the central nervous system. Let me show you a diagram that will be quite clear to you. Okay, here you can clearly see a single oligodendrocyte has multiple process which is wrapping itself around the axons of multiple neurons. While on the contrary, in a single neuron, multiple Schwann cells are forming patches of myelin sheath between which there are nodes of Ranvier. Okay, so I hope it is clear why the central nervous system has fewer nodes of Ranvier compared to the peripheral nervous system. Okay. So this is how here you can see on the outer part there is nucleus and cytoplasm of Schwann cell, while the inner part it's only the plasma membrane of Schwann cell which is forming the myelin sheath. Okay. So this is the step-by-step process of the formation of myelin sheath. So this is how a myelinated neuron looks like. So in the case of unmyelinated neurons, a single Schwann cell can wrap multiple axons. But the difference is that only a single layer of plasma membrane surrounds the axon. So the myelin in the this is a myelinated neuron, while this is an unmyelinated neuron. Okay, so this is myelinated axon and this is unmyelinated axon. So here you can see these are the axons 1, 2, 3, 4, 5, 6. So these are the axons of different neurons, and there is a single layer of the plasma membrane of Schwann cell. So the plasma membrane of Schwann cell does not form a multi-layer structure in the unmyelinated axon. So this is what we call neurolemma. So this is the neurolemma, but it is not forming the myelin sheath. So the unmyelinated axons, they have neurolemma. So because what is neurolemma? Neurolemma is the outer covering of the which is formed by the Schwann cell. So it is the sheath of Schwann cell which has the nucleus and has the plasma membrane. Okay, so neurolemma is present in unmyelinated axons, but the multilamellar structure of the plasma membrane of Schwann cell, which is formed in myelinated axons, is absent. So myelin sheath is absent. Okay, let me write it here. In the unmyelinated axons, myelin sheath is absent, but neurolemma is present. So what is myelin sheath? Myelin sheath is the multi-layered structure. It is the multi-layered plasma membrane of the Schwann cell, while neurolemma is the cytoplasmic outer covering, which even includes the nucleus of Schwann cell, okay, which includes nucleus of Schwann cell. So it is the outer covering which is formed by the nucleus of Schwann cell and the cytoplasm of Schwann cell. Here you can see there's the nucleus of Schwann cell, and then there is the there's the plasma membrane. Here you can see, okay, plasma membrane of Schwann cell, which is forming only a single layer around the axons. So this is the difference between unmyelinated axons and myelinated axons. So this is how a Schwann cell wraps itself around the axons and forms the myelin sheath. While in the oligodendrocytes, a single oligodendrocyte can wrap itself around the multiple axons. Okay, so but unlike that, in the peripheral nervous system, a single Schwann cell will be wrapping itself around the axons of single neuron. Okay, but here you can see in case of unmyelinated axons, a single Schwann cell can wrap multiple axons because it is not going to form myelin sheath. But whenever it is going to form myelin sheath, so in the case of myelinated neurons, in the case of myelinated axons, a single Schwann cell will be wrapping itself around a single axon. Okay, I hope this is clear to you all. So here there's one more diagram. Here you can clearly see this is the axon, okay, and this is a Schwann cell, and this is the nucleus of the Schwann cell, while this is the cytoplasmic part of Schwann cell. So the cytoplasmic part is on the cytoplasmic as well as the nucleus part is on the outer side, and that is why this is what we call the neurilemma. Okay, while this yellow part is known as myelin sheath. So this is the plasma membrane which of the Schwann cell, which is wrapping itself around in the in multiple times around the axon. So this is about 1 millimeter thick. Okay, so this is myelin sheath. So here you can see this is a single Schwann cell. Okay, this is wrapping itself around the axon, and then there will be a gap where the myelin sheath will be absent, and that is known as node of Ranvier, and then another other Schwann cell wraps itself around the axon. So this is how a myelinated axon looks like. And these are multiple unmyelinated neurons, okay, unmyelinated axons around which a Schwann cell is wrapping only a single time. Okay, so there is only single layer of the plasma membrane of Schwann cell, which is known as neurilemma. Okay, so neurilemma is present in both myelinated and unmyelinated axons, but the myelin sheath is absent in unmyelinated axons, and that is why they are known as unmyelinated. Okay, so this is how PNS axons looks like. Okay, so there's one more cell in the peripheral nervous system, the glial cell known as satellite cell. It wraps itself around the ganglia. Okay, so let's discuss about the some terms which are used interchangeably in neurobiology. So what happens is that we already know that in the nervous tissue, the neurons forms connections. Okay, but the neurons, the different part of neurons in the nervous tissue, they are grouped together. So the cell bodies of neurons are grouped together, as well as axons of neurons are grouped together. Just like in muscles, the muscle fibers are grouped together to form the bundles of muscles, right? In same way, in nervous system, the cell bodies are bundled together. They are clustered together, as well as the axons are clustered together. And we have some terminologies to differentiate between them. So let's learn about those terminologies. So let's learn about collection of nervous tissue. What do we call them? Okay. The very first term is first let's learn about the clusters of cell bodies. So when the cell bodies cluster together in the peripheral nervous system, that cluster is known as ganglion. Okay, I've already told you about ganglion in the previous lectures. So the plural is ganglia. Okay, so the ganglia are the clusters of cell bodies that are found in the peripheral nervous system. So these are mainly found in the cranial nerves, in the spinal nerves, around them, around the cranial nerves and around the spinal nerves. Ganglia are they are found. Okay, while if the clusters of cell body they are present in the central nervous system, then they are known as nuclei. Okay, the singular name is nucleus. The singular is nucleus, while the plural is nuclei. So, if we simply call them nuclei if it is found in the central nervous system. Then there are the clusters. Okay. Mm. Then there are clusters of axons. Okay, and if that cluster is found in peripheral nervous system, then we call them as nerve. Okay, so neuron is a single cell. Okay, and neurons are the cells. While when they come together, the axons of the neurons they come together, then they form a nerve. So nerve is a fiber, right? Nerve fibers are means that the axons of the neurons they are coming together and they are forming a bundle. They are forming a cluster in the peripheral nervous system. So we have cranial nerves, we have spinal nerves. So basically, they are not the cell body; they are just the axonic part of the neuron. Okay, I hope that is clear. So bundle of axon. So let's not call it a cluster. Let's call it a bundle. So bundle of axon, which is found in peripheral nervous system, that is known as nerve. While the bundle of axon, which is found in central nervous system, that is called tract. Okay, so whenever I will, I'm saying tract, I'll be referring to the bundle of axons that is found in central nervous system. So those tracts, they are kind of interconnecting axons. They are the interconnecting neurons in the spinal cord and brain. So in the spinal cord and brain, you need some connection, right? So that axonic connection is known as tract. And from the brain and from the spinal cord, there will be arising some bundles of axon which we call as nerve. The bundle which is arising from brain is known as cranial nerves, while the bundles which are arising from the spinal cord they are known as spinal nerves. So I hope those terms are very clear to you all. Okay, so this ganglia, so in the ganglia, the cell bodies of the ganglia, that cluster that is covered by a cell, a neuroglial cell for the purpose of providing protection and support to that cell body cluster. That neuroglial cell is known as satellite cell. So satellite cell is the neuroglial cell of the peripheral nervous system, which wraps itself around it protects and wraps itself around the ganglia, the collection, the cluster of the cell bodies, which is found in peripheral nervous system. I hope this is clear to you all. So, okay, yeah. So there's one more. There are two more terms: the gray matter and white matter, which are concerned about the myelin sheath. Okay. So whenever there's a fresh dissection of brain and spinal cord, when they are dissected, so let's say that this is dissected spinal cord. So there will be a clear, okay, so there will be a clear distinction in the like there will be two regions, a gray region and a white region. Okay, so this, let me, so this is the cross section of spinal cord. Okay, so this is freshly dissected spinal cord, and here you will see a whitish part in the periphery and a grayish part. This is gray part. This is white part. Okay, so this gray, so this gray part is known as gray matter. Okay, this is in the periphery, while this sorry, this is in the this is located centrally in spinal cord, and white part this is located in peripherally. This is located in periphery of the spinal cord, that is called white matter. So the myelin sheath, it is the one which forms the white matter. So myelin sheath, it is made up of lipids and proteins, right? So it looks white, and that is a glistening region which we see in the, so whenever white matter is concerned, there will be bundles of axons. So white matter has axons in that, because not any axons, the myelinated axons. Okay, because myelin sheath is the one which gives the white appearance to that region. So myelinated axons form the white matter, while the gray matter has unmyelinated axons. Okay, it has the cell body part, it has the dendritic part, the dendrites, as well as the axon terminals and neuroglial cells. Okay, and this gray matter appears gray because it has Nissl bodies. Okay, so this Nissl bodies, okay, Nissl body. Okay, this is a new terminology, right? So Nissl bodies, they are the free ribosomes and the clusters of rough endoplasmic reticulum, which are found in the neurons. Okay, so Nissl bodies, they are the free ribosomes and the patches, the clusters of the rough endoplasmic reticulum. So they appear green color, and that is why they give the grayish shade to that region. So the grayish part is present centrally in the spinal cord dissection. But when you dissect brain, so let's say that this is a brain that let me just scroll down. So this is a cross section of brain. Let's say this is a cross section of brain. I know. This is cross section of brain. So in the periphery, there will be a lining of gray matter, and then here and there some patches of gray matter. Okay, so this and the rest of it will be white matter. Okay, so this is the white matter. Okay, so in the spinal cord, when you look at the cross section of spinal cord, white matter will be on periphery and gray matter will be located centrally. While in the brain, white matter will be located centrally and gray matter, there will be a thin lining of gray matter on the periphery. Okay. So in the gray matter we have okay, in the gray matter we have the cell body part. So what, except unmyelinated axons, whatever is there, it is going to be consisted in the gray matter, while in the white matter there will be myelinated axon, because only myelin sheath gives the white glistening appearance to the tissue. Okay, so white matter has the myelinated axons, so all of the bundles of the myelinated axons will be found in white matter, while in the gray matter there will be unmyelinated axons, the cell body part. Okay, so there the clusters of cell body. Okay, so clusters of cell body means the nuclei, they'll be there in the gray matter. Then the axon terminals, the neuroglial cells, all of them except the myelin sheath, whatever which does not has myelin sheath, that will be found in the gray matter. So with this, let's end the lecture here. In the next lecture, we will learn about the electrical signals of neurons. Till then, see you. Bye bye.